Okay, so today we're going to talk about muscle metabolism a little bit, which is really the same as any cell in the way that the cells do metabolism, except for a couple things. But we'll get into that a little bit, and then yeah. So this is kind of we're really moving on from learning all of the specific muscles themselves. Obviously, you're still learning those in the lab, but as far as lecture goes, we're done with the study of naming and identifying muscles here in the lecture part of class. And we'll go over, there's still some people that haven't taken the exam yet, so I won't go over any of those questions until Wednesday's class. But yeah, so we got a couple more days on muscle physiology, today and Wednesday, and then Friday we move into the nervous system to start the nervous system. Okay, so let's start out here. Start with the end in mind. What is the end goal of all metabolism in the body? Go ahead, talk with your team about this. Oh. Okay. That's what it's All right, we got 49 so far. Go ahead and send it in if you haven't yet. There we go, 59, 60, 63. Anybody else? Is that everybody? All right. One thing on the iClicker points. Right now, the last time I updated it, which was before the exam on Friday, that's when we finally got to being out of 40 points, which means like that's the total that we're going to get to in that column. So if for some reason you were behind from the beginning of class, or you didn't have your clicker for the first couple of days or things like that, then now's the time where you'll start catching up. Okay, so if you're so that's how it works. And so if you're already at 40, then you're at 40. You're not going to get any higher than 40. Okay, but otherwise you get 2 points per day for using it, and so you'll start catching up now all the way until the end of the semester. Okay, so don't freak out if you're a few points behind on that. You'll most likely catch up. Okay, all right, let's see, take a look and see what you guys said here. Okay, so 71% of you said C, to turn ADP into ATP. Okay, and obviously this term metabolism is used kind of real specifically here to talk about this, but it could be used to talk about other things, breaking down things in the body. But the way we're talking about it today is breaking down food sources, so specifically macro nutrients, and turning them, using them to make ATP. Okay, and so we're going to talk about the 3 energy systems that we find in skeletal muscle cells today. And so here they are. So the 3 are, the first one is called the creatine phosphate system. Okay, this is definitely the fastest one. Gets us ATP immediately. There's really only one step in the reaction. Okay, there's no food-based substrate needed for this. This doesn't use carbohydrate, protein, fat, alcohol. It doesn't use any of those substrates. Okay, it gives us the least amount of ATP. Only gives us one at a time. Okay, but it also has it wears out the quickest. Okay, when we're doing a full-on sprint, we would deplete this in about 10 to 15 seconds. Okay, the kind of middle one is called the anaerobic pathway. Okay, the anaerobic pathway is kind of our second fastest, our second in terms of fatigability. Okay, this one requires glucose or glycogen. For the most part, though, we're going to be using glycogen. It's also known as the glycolytic system. Okay, it's just breaking down of glycogen. Okay, for this one, we get 2 ATP per glucose used. When we use glycogen, we get 1 more ATP than that. This one wears out in about 1 to 2 minutes of an all-out type sprint. Okay, And then there's the third one called the aerobic pathway, which is what the vast majority of all of our ATP is made through. Okay, this one's definitely the slowest to get us ATP. This is the one that we still can use glucose, but we can also use fats and proteins to make energy here. Okay, this one gives us around 32 ATP per glucose. Okay, and one note about this, you may have gotten different numbers on the number of ATP you get from this in different classes. This is not like a set thing at all. That depends on the state of the mitochondria and different things going on. It does seem to be lower than we once thought. We used to think it was somewhere around 39. It's probably not quite that high, but just for this class, this is the number that I want you to know for this. Right? And this one, the fatigue part of this is really only limited by the amount of substrate available. And for the most part, we pretty much never run out of substrate for this one, because we all, even if we're really skinny, we still have a lot of fat to use. Okay. All right, so those are the three energy systems, and we'll talk about each one with a little bit more detail here right now. Okay, any questions so far on this? All right, so we'll start with the creatine phosphate system. What we have inside of our muscle cells are these creatine proteins, okay? And what they do is they hold on to a phosphate molecule for us, okay? And really with the help of this enzyme called creatine kinase, the creatine phosphate molecule, which is shown there, the creatine plus the phosphate, with the help of that creatine kinase, takes that phosphate, takes it off of creatine, and puts it onto ADP to turn it into ATP, right? So then we go from having a creatine phosphate plus an ADP to having just a creatine plus an ATP, right? 3 phosphates instead of 2, right? So that's really it. So it's really just that one-step reaction, super fast. And so really what happens is when we start moving our body, right? Let's say we're just standing here and all of a sudden I just start walking really fast, okay? All of a sudden the ATP that's stored in my muscle cells is going to get broken down. It's going to get turned into ADP and a free phosphate, okay? And so when those, so by the presence of having a bunch of these ADPs floating around, this stimulates this process, and then that creatine phosphate can just put a phosphate right back on ADP, turn it back into ATP, and it's ready to be used again, right? And so it's just a super quick replenishment of that ATP that got used up. Okay, so very fast, very high energy production. So some type activities that would really exemplify the use of this would be like really short sprints. Okay, so like a 50 meter dash or something like that, 100-meter dash. Something that's lasting 10 to 15 seconds would really exemplify the use of this, but even more so would just be like a single movement, like swinging a bat as hard as you can, or jumping as high as you can, or any kind of power type movements. That's really where you're going to really rely on this system primarily, more than any of the other 2 systems. And so this one applies directly to a very popular supplement, the creatine supplementation, right? You guys have all heard of creatine supplementation, something you can like mix up in water and drink, and you're really just adding more of that creatine to your muscle cells if you're doing it right, and then you just have more ability to have that stored energy source. And so really what creatine supplementation does, it gives you a little bit more energy to push yourself a little bit harder. It doesn't make your muscles get bigger. It's not anabolic like that. It just gives you the ability to push yourself a little bit harder. So then you have the ability to put a little bit more of a stress on your body, and then your body can adapt a little bit more than it normally would, because you put more of a stress on it, right? And creatine supplementation is one of the ergogenic aids, what it's called, things that are supposed to help you perform better. That actually does work. There's actually solid research around creatine supplementation that shows it makes you be able to work out harder, for sure. Okay. Yeah. Any questions about any of that? All right, so that's the first one, the creatine phosphate system. Now we'll talk about the other 2, and both of them start with glycolysis. Okay, so this is kind of the common pathway for the next 2 systems. And glycolysis has a bunch of steps in it, as you can see here. You don't have to memorize each one of these steps for this class. Okay, maybe if you take another class later, you'll have to get into some of the details here. But really, what we need to know about it here is that we're going from glucose to pyruvate. Okay, for this class, we can take a shortcut from step one to the last step. Okay. And that's really what you need to know about glycolysis. We're going from glucose to pyruvate. Yeah, and so that's it. So what we see here, glycolysis, the common pathway, the breakdown of glucose to make pyruvic acid, or also known as pyruvate. Okay, this happens in both the aerobic system and the anaerobic system. Okay, if we're starting with glucose for the aerobic system, but the aerobic system can also use lipids and proteins. If we're doing that, then we don't need to do glycolysis first. But if we're starting with glucose, which is the one we'll focus in on today, then we're going to start with glycolysis no matter whether we're using the aerobic system or the anaerobic system. Okay, so it can go from glucose. It can also start with glycogen. Right? What was glycogen? What is that? What is it? What kind of molecule? How about that? See, I feel doing stuff like this. What is it? What kind of molecule? What kind of macro or what kind of organic molecule? It's a, say it again? Polysaccharide. Okay, good. So it's a carbohydrate, polysaccharide, multiple monosaccharides strung together. This is just a bunch of glucose molecules strung together. Right? This is our stored form of carbohydrate in the body. Okay, it's really our only way that we store carbohydrate. If we just leave it as glucose, we don't really store much of it. So we can take the glucose, put a bunch of them together, make glycogen, and we can store it. Okay, the two primary places we find it stored are in our liver and in our skeletal muscle. Okay, those are the two. Way more in the skeletal muscle than anywhere else, but we also have it in our liver. And so it's a stored carbohydrate. Okay, so that's what we can start with, either glucose or glycogen. And then the other things that we get out of, besides the pyruvates, so we get this out of glycolysis, but then we also get a couple of ATPs made here during glycolysis. And then we also get a couple of these NADH molecules. And we'll talk about the importance of those here very soon. Okay. All right, so that kind of goes from glycolysis, will get us down to pyruvates. And then the other key thing here is which direction is pyruvate going to go after we make pyruvate, okay? And so we go from glucose to pyruvate. Here's our glycolysis. And then at this point, there's kind of a fork in the road. We can either go the aerobic pathway, and that would be turning pyruvate into acetyl-CoA, or we can go the anaerobic pathway. That would be turning pyruvate into lactic acid. Okay, we don't do this one, but certain microorganisms do, right? Like fungus and certain bacteria. They can turn pyruvate into ethanol, which is alcohol, which is the exact process used by yeast to make alcoholic beverages, right? It's exactly how ethanol, which is alcohol in beer, wine, hard alcohol, is made through this process, right? But humans don't have that third option. Well, the microorganisms typically only have that option at times, but yeah, so we can either do the aerobic pathway or the anaerobic pathway. Okay, so then it's really the fate of pyruvate or the direction it goes is really going to determine whether we're going to use the anaerobic pathway or use the aerobic pathway. Okay, so let's talk about the anaerobic pathway first. Okay, so again, we're going glucose to pyruvate first. That's our first step. And then we end up with 2 pyruvic acids. Okay, and then that's going to eventually turn into 2 lactic acids. All right, so we can see that here, pyruvic acid down to lactic acid, and then that lactate can be released into the blood. Okay, so the things we get out of this are the things that we've already seen. We got the 2 ATP from glycolysis, we got some NADH from glycolysis, and those are the 2 key things. Okay, so no oxygen is used to do this. There's no oxygen in this pathway. That's why it's called the anaerobic pathway. Okay, so this gets us energy pretty rapidly. Okay, but it's also an extremely inefficient use of glucose and just kind of a waste of energy in our bodies. Right? So you think about this glucose only is giving us 2 ATP, whereas when we do it the aerobic way, then we get 32 ATP out of it. Right? So this is just a total inefficient use of energy, but sometimes it's necessary to get energy really rapidly, And that's really what this is all about. The theta pyruvate is dependent on how fast we need energy. If we need it really quick, then the body's going to send it this direction, and we're going to get more ATP, or we're going to get ATP quicker. And so this would be like if we start sprinting, we're running, we're going to need to use this system more so if we're running fast versus if we're just walking, because we need that ATP rapidly. Okay. So where in the cell does this one occur? Do you guys know which part of the cell? No, not the mitochondria. This one happens out in the cytosol. Yeah. The aerobic system, most of that happens in the mitochondria, but not this top part of it, not the glycolysis part. Okay. Then we have lactic acid. Okay, so lactic acid is what comes out of this along with the ATP and the NADH. Okay, like any acid, it can release a hydrogen ion, right? It can donate a hydrogen ion to a solution. Okay, but then what happens with that is that hydrogen ion can bind to things like pain receptors, and it can cause pain, right? So this is really the source of the burning that you get during exercise. When you're doing an intense bout of exercise, you start feeling some burning in your muscle. This is really the source of that, is that drop in pH that occurs by doing more glycolytic metabolism or more anaerobic metabolism, and then that lactic acid releasing that hydrogen ion, dropping your pH and stimulating pain receptors. Okay, it's also a negative feedback system to where those hydrogen ions don't just bind to pain receptors, they also bind to all the enzymes involved in that whole process of glycolysis. And when they get enough of them bound to them, they don't really do their job well anymore. They start unfolding a little bit. And so then the system that produced the lactic acid is also limited by the lactic acid, okay? Because it messes with those little enzymes that are doing all of that. And so then we don't get as much, we can't really use that system as much. So it's kind of the self-limiting thing, which is good because we wouldn't want to be able to drive our pH too low, because that could hurt us. Okay, but there's been a lot of stuff over the years about lactic acid. It's kind of been blamed for many things, including delayed onset muscle soreness. Right? Some of you probably had a coach in high school tell you like, oh yeah, if you're really sore, then go out for a run. You know, you're sore the next day or a couple of days after workout, go out for a run, get all that lactic acid out of your system. Has anybody ever told you guys that? Yeah. The problem is all the lactic acid is out of your system within an hour after exercise. Okay, so that's an old paradigm. It's still a good idea to go for a little jog and clear out some of the stuff that's causing the pain, but it's not lactic acid. It's inflammation that's caused by the workout that you did. And so the lactic acid is not really the cause of that. What happens to lactic acid is a couple different things. Number one, it can get into the bloodstream, get circulated to the liver, and then the liver has a special process that it can just turn it right back into glucose. Okay, so that's one option for it. Okay, another option is, and the most common option is that when that lactate is made, it's either used again right there in a muscle cell that's close to that one that it was made in the same muscle cell, or it's actually used very heavily in the heart, in the kidneys, and the brain too. I should have the brain on here. The brain is a large user of lactate, and by using it, what I mean is, if it was only one step to go from pyruvate to lactate, then that reaction can just go the other direction too, right? We can just turn that pipe. We can take that lactate, send it right back through that same enzyme, just the opposite direction, turns it into pyruvate, and then it can go the other direction to acetyl-CoA, and we can do the oxidative system. Okay, so it's not really this like terrible byproduct that we once thought it was. It's actually a really useful source of energy, and the body uses it a lot, uses it very heavily. So almost all of the lactate, something like 75 to 80% of the lactate that we produce is just used again through oxidative metabolism, either by the muscle cells or the heart or the brain. So yeah. All right, so then this, like I said, this can cause the burning. But that pH change that we get is going to be gone most likely within 30 minutes of an intense workout. Our pH is going to be back to normal. Definitely within 2 hours, the pH and the lactate is going to be all back to normal. So nothing to do with that soreness we get a couple days after a workout. Okay, any questions on any of that? All right, let's go ahead and do this question. All right, go ahead and send it in if you haven't. Try about 10 more people. All right, a few more people. Make sure to send it in here. Now that I told you guys, you got the full points. Maybe some people aren't going to vote now. No, I'm just kidding. All right, we're good. We got it. Okay. All right, good. So 89% of you are saying C. Okay, whether the aerobic or the anaerobic system is used. Okay, absolutely. So pyruvate goes to lactate. That's the last step in the anaerobic system. We're done. If it goes to acetyl-CoA, then we're going to really start the unique parts of the aerobic system, and that's where we'll hop into next. All right, questions on any of that? Okay, so this is kind of the broad overview of the aerobic system. Okay, so we first start with glycolysis, and this is the aerobic system when we're talking about carbohydrate metabolism. Okay, start with glycolysis, then we go to the citric acid cycle, also known as the TCA cycle or the Krebs cycle. Okay, and then we go to the electron transport chain. Those are the three kind of big steps in the aerobic system. Okay, so let's talk about those steps here. All right, so again, steps if we're breaking down carbohydrate, which is the only one we're really looking at for this class, then we're going to start with glycolysis. Okay, there's another step in there before we get to the Krebs cycle, and that's just turning that pyruvate that we made in glycolysis into acetyl-CoA. Okay, this doesn't have like a big name like the other ones, but it is that step between pyruvate and acetyl-CoA. And then if we get to acetyl-CoA, that's what we need to start the Krebs cycle. Okay, and then from the Krebs cycle, we move to the electron transport chain, which is also known as oxidative phosphorylation. Okay, so we'll talk about each of these in a sec. So the overall products / byproducts, we make the majority of ATP here. Okay, so 32 ATP total with all those steps, and we'll see where each of those ATP come from in a sec. Okay, this is where we get the CO2 that we breathe out from a couple steps along the way. And we get quite a bit of water this way. It's called metabolic water production. Happens in the electron transport chain. Okay, so we can either use carbohydrates to do this, is one we'll talk about. We can use lipids, or we can use amino acids from broken down proteins. Okay, so this is kind of just looking at those all the different macro nutrients that we can use in the aerobic system. All right, so carbohydrates is where we'll spend our time talking about today. Okay, but we can also use lipids. We can also use proteins. Okay, with proteins, we need to break them down into their individual amino acids first. And then the thing about the amino acids is they all kind of jump into the aerobic system in different places. Okay, some of those can be turned right into pyruvate. Some of those can be turned into acetyl-CoA. Other amino acids can be turned into other steps along the Krebs cycle here. Okay, we won't get into the details of that for this class at all. Okay, and then with fats, we break fats down into fatty acids and glycerol, go through a couple different steps, but we still end up producing acetyl-CoA, and so we still use that same acetyl-CoA pathway that we do. We just have to get it down to acetyl-CoA first through some other steps. Okay, so again, we'll focus in on the glycolysis part here, the carbohydrate part. Okay, this is just kind of an overview of the Krebs cycle. Again, for this class, you don't need to memorize this. Okay, there are some other classes where you might need to, like some of our nutrition folks, I know need to memorize this. But yeah. Okay, so the key things with the Krebs cycle that I want you to know are, number one, what it starts with. It starts with acetyl-CoA. All right. So each mole of glucose will give you we give you 2 moles of acetyl-CoA. All right, and then with each acetyl-CoA, we get 1 turn of the Krebs cycle. So that means with each glucose, we get 2 turns of the Krebs cycle. It goes around twice. Okay, and then the things we get out of the Krebs cycle are, we get a bunch of NADH, which is going to be an important hydrogen/electron carrier. We get some FADH2, which also plays that same role for us. It carries electron-rich hydrogens to the electron transport chain. And then we get 2 more ATP made right here. Okay, we're also going to get some CO2 here as well. That'll be more of a byproduct. Okay, so a little bit more about these hydrogen/electron carriers is NAD+. That's before we put the electron-rich hydrogens onto it. Is niacin, which is vitamin B3. Okay, so this is one of those key vitamins that we have to have taken in through our dietary intake to be able to do just normal life. But if we don't have niacin, we can't do this. We can't really function for very long. Okay. Same thing with FAD. This is another B vitamin. This is riboflavin, so vitamin B2. Okay. Without vitamin B2 and vitamin B3, we don't live for very long because we can't really do the electron transport chain anymore. We can't really make much ATP, and so these are absolutely essential for life. All right, so got a brief video here, just kind of the overview of the electron transport chain. So I'm going to go over to YouTube here, and the sound's going to work. It is, because I tested it. I'm willing it to work by saying that. Let's make sure it does. Okay.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
Okay, so that was just a kind of a nice overview of the electron transport chain and how it works. And so you saw those NADHs and those FADH2s kind of bringing electrons to the inner space of the mitochondria. Okay, and yeah, so we'll walk through a little bit of that. So there's a lot of other steps in here, but we're simplifying it for here. And so here's the things that I want you to know about the electron transport chain. Okay, number one, this is the last part of the aerobic pathway. Okay, so we started with glycolysis, went to the pyruvate to acetyl-CoA, Krebs cycle, and then electron transport chain. Okay, one of the key pieces in understanding how this works is that it uses secondary active transport. Okay, so when we talk about secondary active transport, that means we're having to move something in which direction? With its gradient or against its gradient? Against. Okay, active is what tells us that. The active part tells us that we're moving something against its gradient. Okay, and then if it's secondary active, what is it that powers us moving something against its gradient? So not ATP. So primary active transport is when we use ATP. Secondary active transport, we use the gradient of something else, right? So we're kind of like hitching a ride with something else that has a gradient to move in a certain direction, and then pulling along that thing that doesn't have the gradient to move in a certain direction. Okay, so the way this works in the mitochondria is that those electrons, and we'll come back to this in a sec, but those electrons are dropped off here. Okay, and we can see those dropped off. They stay within the membrane by getting bound to different proteins, and they flow this direction. Okay, so they flow down the membrane, and they have a strong electrical gradient that pulls them that direction. Okay, so they're going with their electrical gradient. Okay, and there's energy in that, right? And the energy in that is used to kind of sling those hydrogen ions against their gradient, right? And that's the secondary active transport part, is that we kind of harness the energy of those that electron gradient to kind of grab a hold of those hydrogens and throw them across. Okay, I think the best way to think about this is like a catapult, right? You think about like a catapult in like in a movie that's like showing like old like Roman times or something, and there's this big arm like a teeter-totter, right? One side's way down here, one side's way up here, and maybe there's like a flaming ball or something down here, right? And then there's somebody way up high, and they can drop something on that other arm, right? And it flings that flaming ball across some wall and knocks a bunch of stuff over, right? Starts a big fire. And so the thing falling to do that, that's the electrons, right? The electrons are using gravity pretty much. Not this, it's not gravity, but the ball is using gravity to fall. And then that takes the flaming ball on the bottom that doesn't have any potential to go up unless we use some energy, and it flings it across, right? So that flaming ball is the hydrogens. The ball falling from up high, or the rock falling from up high, is those electrons, right? So we're taking that power from the electrons and flinging the hydrogens across. Okay, so we end up with a ton of hydrogens up here, right? And that's the whole point of those electrons, is so we can build up this strong concentration of hydrogen ions in that intermembrane space. Okay, and then there's only one of these proteins that lets the hydrogens flow through, and that's this one at the end called ATP synthase. Okay, and those hydrogens rush through that ATP synthase, and they turn this giant thing. And it actually has all these components of a rotary engine, very similar to a rotary engine that humans have made. And it turns this big old thing and really mechanically just smashes the ATP or the ADP and the phosphate together and makes ATP. Okay? And so we needed to make that gradient first by the power of the electrons, and then we use that hydrogen gradient to move this big ATP synthase and make the ATP. Okay, so that's really that. So the key in understanding here is that we're using secondary active transport to create a gradient for those hydrogen ions. Okay, we use that gradient to make our ATP. Okay, talked about all that. I got super excited, and we haven't even talked about oxygen yet. Oxygen's a part of this, right? That's the whole reason we breathe. Okay, oxygen's the final electron acceptor, right? And the reason that's so important is that if we didn't have anything to grab onto the electrons at the end, then they would all build up down there, and we wouldn't have a gradient for the electrons to go that direction anymore, right? We don't have a rock to drop from the top, and we can't throw that flaming ball over the wall. And that's exactly what would happen, right? If we just let the electrons build up, then the whole thing stops and we can't move hydrogen ions anymore, right? And so it's the same idea. So we need to have something to bind to those electrons, which is oxygen. And we really use that to make water. And then we can keep sending electrons down because we maintain that strong gradient there. Okay, so oxygen's final electron acceptor. 28 of the 32 are made right here. Okay. So the majority of them are made right there. Okay. Questions at all about that process? All right. So we kind of already walked through this. The one thing I'll point out here is that we didn't really walk through that part. So we got 2 electrons there, right? And that's the end of the electron transport chain. We got half of an O2, which is just an O, right? And then we combine that with 2 hydrogen ions, and that makes water, right? That's how we get water. So that's how we get water at the end of the electron transport chain. Okay, and again, that's that important step of maintaining that electron gradient right there. Okay. All right, so just take a second with the people on your team, add up the ATP. Where did each of those come from? And you can send in the number there on your clicker just to make sure we're all on the same page. So go ahead and do that.
What is that? What is it? What is NADH?
So it's a that would be the same. That would be the guys in the plastic box. Yeah, I carry the rocks over to the catapult. Yeah, it could be, doesn't work quite as well. So I have a picture of it, like, sure, yeah.
Is that the light or something?
It is.
That's like always. I always get my voice.
You're gonna hear this, right? All right. Make sure you hit send on this type of question, guys. Hit the send button. So you're putting in the number, right? And hit send. All right, I think we just about got everybody. Okay, so so pretty much everybody, except for one person at the end there that just put in a bunch of twos to get something in. Put 32. Okay. All right. And then so how many do we make in glycolysis? 2. Okay. How many in the Krebs cycle? 2. Okay. And then how many electron transport chain? 20. Okay. Good. All right. Okay. Another quicker question here. Let me just reset that. So back to multiple choice. Okay. Go ahead. And you can talk about this question and the next question with your team, and we'll just vote for them both sequentially. All right, put in your answer for this one here. All right, everybody make sure to put your answer in here. Just press it again to make sure we got 56 so far. All right, anybody else for this one? Okay. All right, so everybody is pretty much saying C, so to accept the electrons at the end of the electron transport chain. Good. Okay, I'm going to open it up again. You guys should be all ready with this answer. Go ahead. Go ahead, put in your answer for this one. Which order of events is below is correct for the oxidative system when using glucose? All right, anybody else? Is that everybody? Okay, we're good. All right. 100% of you are saying D, glycolysis, pyruvate to acetyl-CoA, Krebs cycle, electron transport chain.
So true.
Good. Okay. All right. One quick story about ATP, then I'll have you guys do this recap. That conference that I was at a couple weeks ago that you guys that I wasn't here for. There was a talk on that from one of the world leaders in understanding exercise metabolism. He did an hour-long talk on how much ATP is used during the 2-hour marathon. Okay, so the 2-hour marathon has it's only happened once in the history of humanity, as far as we know. It was by the world record holder in the marathon right now is a Kenyan, Eliud Kipchoge is his name, and they had this whole Nike project called Breaking 2. You guys, anybody seen that? There's a documentary on Disney Plus. You should check it out. It's pretty fantastic. Kipchoge is a really cool guy as well. But they pretty much tried to set up everything perfect for him to be able to break the 2-hour mark, because it has never happened before. I won't tell you the end result of the movie, but eventually he does break the 2-hour thing. But pretty much from all of the data they have, they know exactly how much ATP he's using. And so this whole talk was trying to figure out how much ATP is used during a 2-hour marathon. The end result is completely mind blowing. To do that, you have to burn 56 kilograms of ATP in 2 hours. He only weighs probably 60 kilograms. And so the amount of energy that is required to do that is 56 kilograms. Okay, and so they're taking in lots of fuel the whole time, but he's also much lighter afterwards as well. But yeah, just to put it into perspective, the pace that you have to run at to run a 2-hour marathon is like a 4:49 or 4:48 pace, something like that. He's almost running. It's like almost 14 miles an hour for 2 hours, right? Yeah, so they're just kind of superhumans for sure. But yeah, pretty mind blowing. Yeah. So it's incredible. But with that in mind, go ahead and do this little recap here, and we'll wrap up with this. This is going to be like ranking them, like fastest, slowest, in the middle. It's just
Super fast. Middle. Wait, this one's because for anaerobic, this one, yeah. It's like a chipmunk. Okay, not a lot. Four. Yeah.
All right, guys, let's come back together and just fill this in real quick here. So, which one's the fastest energy system? Okay, so I'll just go up, down, and middle like that. Okay. Substrate required. What do we need to do the creatine phosphate system? We need creatine. But we don't need carbohydrates, proteins, or fat, but we do need creatine. Okay, anaerobic pathway, which is the primary substrate for that? Glucose or glycogen. Okay, this one uses glucose, it uses lipids, it uses protein. Okay, number of ATP creatine phosphate system, one at a time, anaerobic, 2, aerobic, 32, okay? And then the endurance one, which one has the most endurance? Okay, so it's just gonna be the opposite of the speed, middle, and lowest. Okay? All right, guys. Have a great rest of your day. See you on Wednesday.